Hi, my name is Tad Wanveer. I'm the developer of CranioSacral therapy, Touching the Brain 1 and 2. This is a brief overview of the material covered in the first class, CranioSacral therapy, Touching the Brain 1. Thank you for joining me. 50% of the cells in the central nervous system are glial cells. Therefore, an understanding of glia can be helpful in our work as craniosacral therapy practitioners because much of our focus is upon the structure and function of the brain and spinal cord. I think this quote sums up the importance of glial cells. Glial cells are central nervous system homeostatic cells. They regulate all aspects of central nervous system function in physiological and pathological conditions. Neurological diseases should be considered as gliopathologies, which determine the progression and outcome of neuropathological processes. So if we were to summarize this, we could say that glial cells are the homeostatic cells of the central nervous system. And neuropathology appears to be based in gliopathology. So it can be helpful for us to have an understanding of glial cells. The material covered in class will be first, we'll work with drainage pathways. In every organ, waste clearance is as basic an issue as how nutrients are delivered. So therefore, we'll work with 7 primary drainage pathways. And part of that work will be working with production of cerebrospinal fluid at the choroid plexuses, flow of cerebrospinal fluid into the brain and spinal cord, blending with interstitial fluid, picking up waste products, flowing back out into the subarachnoid space, and then to eventually enter into 7 drainage pathways. So we will work with these pathways, and then we'll move on to work with a system that feeds those drainage pathways, and it is called the glymphatic system. It is a system that regulates the inflow of cerebrospinal fluid and the outflow of interstitial fluid. The outflow of interstitial fluid cleanses the central nervous system of harmful substances. From here, we will talk about that system in detail, and it's comprised of what's called astrocyte end feet that encase the blood vessels. End feet are portions of astrocytes. Astrocyte means star-shaped cell. It's a form of glial cell. We'll also work with the blood-brain barrier, which regulates the flow of blood-borne substances into the interstitium. We'll also work with a system called the neurovascular unit, which regulates micro blood flow throughout the central nervous system. From here, we'll work with radioglial cells, and radioglia are a form of transient, meaning temporary, glial cell found during in utero development. So we will discuss radioglia and work with them as well through working with their patterning in existing astrocytes. Moving on to then discuss in more detail astrocytes. And astrocytes set up what's called microdomains, little tiny neighborhoods throughout the central nervous system. And they then act as the hub of regulation and integration for the structures that they surround or attach to. And if the astrocyte processes, which are called end feet, are near the pia mater, they attach to the inner surface of the pia mater. And they also interconnect with other astrocytes to form an interconnected astrocyte matrix called the syncytium throughout the central nervous system. They surround blood vessels, forming a portion of the blood-brain barrier and contribute to the neurovascular unit. Also, if they're nearby the ventricular system, the ventricles are created by ependymal cells, a specialized type of glial cell. The projections of astrocytes will adhere to the outer surface of the ventricles. The end feet will also surround axons, oligodendrocytes. Oligodendrocytes are the myelinating cells of the central nervous system. And the astrocyte will also encase neural cell bodies and synapses throughout the central nervous system. So we will work with the interconnection of the astrocyte with a pia mater, the interconnection of astrocytes with other astrocytes, the interconnection of astrocytes with blood vessels, as well as how they adhere to the ventricular system. We will also discuss oligodendrocytes, axons, and work with both. And we will work with synapses throughout the central nervous system. And astrocytes act as the regulatory hub of the processes of the structures they surround within their domain. And from here, we'll move along to work with what's called the three-part synapse. The three-part synapse is comprised of a presynaptic neuron, a postsynaptic neuron, and astrocyte end feet that encase the synapse. And we will discuss the three-part synapse and how an astrocyte helps to support and contribute to the work at the synapse. We'll also discuss and work with the extracellular space, which is a minute complex space through which the substances within the brain and spinal cord travel. So once these substances either leave blood or travel through the cerebrospinal fluid pathways, they'll enter into the extracellular space and they'll be carried by interstitial fluid. So we'll talk about this and we'll work with the extracellular space as well. And then from here, we'll work with four basic types of transmission in the central nervous system. One that we're familiar with is called wired transmission. And wired transmission is the way in which neurons receive, organize, and transmit information. It's considered a one-to-one type of transmission. The other three forms are considered as volume transmission, which means one to many. And they're called extracellular volume transmission, ephaptic coupling, and intracellular volume transmission. So we will discuss and work with all of these four forms during the class. From here, we will work with oligodendrocytes, discuss myelin, work with axon pathways with their myelin sheath, which is referred to as substantia alba. And then moving on to gliosis, which is neuroinflammation. And gliosis is the inflammatory process. The cells involved are called microglia and astrocytes. Microglia are the resident immune cells of the central nervous system. They respond to cell injury or infection, such as bacteria, viruses, trauma, and autoimmune response, dead cells, toxic proteins, harmful chemicals, and aging. And they change their cell structure and processes to meet the demand of challenge, and that activates the inflammatory response. During the virtual class, you will work on yourself. And this is a quote from Melissa, who was in a recent virtual class. She says, "Working on myself versus another student was a profound learning experience. It allowed me to learn each technique in my cells." So we will deepen our work to our own inner awareness, which I believe deepens us to our work when we're in the clinic. And to close, there's a few quotes I like to share. Homeostasis is life's fundamental property. In short, homeostasis is life. The central nervous system operates as the controller of internal homeostasis. Glial cells are the central nervous system homeostatic cells. Therefore, working with glial cells can optimize life's expression throughout the whole body, enhancing self-correction and wellness. Thank you for joining me. I look forward to seeing you in a class, whether virtually or in a live class when we're able to get back together again. I wish you, your loved ones, your family, and those you know, all wellness, happiness, and thank you very much.